Okay, hi. So we're gathered here tonight, or today, this morning, depending on when you're watching this, to talk about a couple immunodiagnostic tests, specifically some primary binding tests. And when we say primary binding, that's antibody binding to antigen directly. So let's just get right into it. So we'll start with some immunofluorescence type things, which are pretty, I think, because you see a bright light. So the first one that we're going to be concerned about is called direct immunofluorescence assay, and I just abbreviated it to IFA. So the first thing we want to know is are we looking for our antigen or are we looking for our antibody? And because this says direct, we know that our antigen is going to be our unknown, so that's what we're going to be looking for. Before we talk about the antigen, I just wanted to briefly go over just a basic diagram of an antibody. So essentially we have an FC region here and we have an FAB region, and the FAB region is going to be the part that will actually bind to our antigen. So these antigens are going to be, so what we have here is we have a slide, and the slide is going to have a tissue section on it. So it could be like any tissue basically. The examples that I provided here, which are from the lecture notes, are some brain tissue, and then to detect rabies, which is a virus, and we also have the bovine viral diarrhea virus, something like that. And basically, viruses, they're intracellular parasites, so what they're going to be doing is they're going to actually go into the cells, and they're going to be replicating in the cells, and as a result, we're going to see an antigen presented on that cell. So basically, we can just pretend that this is our slide. We have a tissue section. We can pretend it's brain, and on the brain, there's going to be some antigens, which are these blue spikes. And so the antigen, that's our unknown. So what we're going to do using this direct test is we're going to use a known antibody, and that known antibody is going to be able to bind to our specific antigen. So what happens here is the known antibody binds to that antigen, and it has something called a FIT-C, which is basically just a fluorochrome, so it's easy for us to identify. And so what happens with a positive test for this is we're going to see fluorescence. If it's a negative test, we're not going to see any fluorescence because this will be unable to bind. So if this antibody doesn't recognize this antigen, it's not going to bind, and we're not going to see any fluorescence. So that's the first part. So that's the direct. So now we're just going to do an indirect version, which is only slightly more complex. So the indirect immunofluorescence assay, in this situation, our antibody is now the unknown because it's indirect, so antibody. So it's the same tissue sample, although it could be from a different virus, or we could even use it for titrations. This is some sort of porcine reproductive virus. And so what happens is we'll take our sample, we'll put the tissue section on the slide, and we have our antigens. So now our unknown is going to be our antibody, and our known is going to be an antigen. So we know what this antigen is. So what happens is we'll put our patient's serum through this, and if there's antibodies in the serum, and when we say serum, that's just whole blood minus the cellular components, so like no clotting factors, no cells. And so what happens is we're going to spin that down, and we're going to get the serum, which is going to be where our antibodies are going to be located in that serum. And so when we run the serum over this sample, if there's antibodies that will recognize this antigen, they're going to bind. And then what we do is we have, so if you see here the FAB region will bind that antigen, and then we're going to use another FIT-C fluorochrome stain that's going to bind to this specific unknown. And if it's positive, then we're going to see a fluorochrome, we're going to see a bright fluorescence, and if it's negative, then we're going to see no fluorescence. So that's basically how it works, and that is the first immunodiagnostic test, immunofluorescence. So let's keep going. Okay. So the next test is ELISA test, which I'm sure a bunch of you are familiar with. It stands for enzyme-linked immunosorbent assay. And so the first one that we're going to look at is our indirect one. That's not right. Oh, jeepers. So for indirect test, our unknown is going to be our antibody. So basically, that's what we're going to be looking for in this situation. So we have these nice little well plates here, and the well plates are going to be coated with a known antigen. So we don't know the antibody, we do know the antigen, and so what we're going to do is we're going to take our patient's serum and we're going to run it over here. So what happens is when we run our patient's serum, if it, and this can be used for titrations, which just will evaluate how much antibody is in the patient's serum. So if it is recognized, then basically the antibody is going to bind to our antigen, and this is going to be an unknown antibody, and then what's going to happen is we're going to have another one of those special antibodies that has the fluorochrome stain on it, and it's going to, oh jeepers, it's really stuck on there. All right, and then so it's going to bind like this, and yeah, so then we're going to see a fluorochrome, we're going to see a color emitted, and we're going to know that it's positive. So then slightly different, but a little bit more confusing, I would say, is going to be our direct. So in this situation, the unknown is the antigen, so we're trying to figure out what that antigen is. And so now, the way I like to think of this is we've kind of just flipped this a little bit. So instead of our well plate being lined with antigen, it's now going to be lined with a known antibody. And so what happens is we're going to run our patient's serum over this sample. And so this patient's serum, these little red dots are indicating heartworm antigen, protein antigen. So what's going to happen is if our patient is positive for heartworm, we're actually going to see these are going to stick to these known antibodies here. And so once they are stuck there, then we're going to run it with the fluorochrome as well. And what's going to happen is it's actually going to become sandwiched. And so that sandwich is then going to give us a color, and we'll know if our patient is positive or negative for heartworm. And so these are going to convert into S, and then we'll see a color. And same thing with this. So every time you see an S and an E, it's going to convert to an S. So yeah, so that's the first two parts of the ELISA test, and like I said before, we can do this for heartworm. We'll just keep moving these whiteboards. All right, and then so the last one, which I think is really interesting, we have a competitive ELISA test. So essentially, we can do this if we're working with wildlife, and we want to know basically, is our patient positive or negative for a specific antibody? But we can't make these antibodies and can't come up with all these antibodies all the time, for all these different species, maybe like a polar bear or something. I don't even know. But so what we'll do is we do something that's competitive. So we have a known antigen, and we line our well plate with a known antigen, and then what happens is so we're then going to run our patient's serum over those well plates. And so what happens, which I think this is really cool actually, but maybe you won't, I don't know. So what we're going to do is I'm going to run my patient's blood, and this is going to be a positive test because this patient, we can just pretend it's a polar bear or something, I don't know, that's kind of cool. And so the polar bear serum is then going to bind to this, which is kind of cool. So when it binds to this, and then we're going to run something with our fluorochrome over it. So once we do that, we're going to actually see that this is going to be no fluorescence. Oh, okay, yeah, so this is cool. So what's going to happen is we're going to run the fluorochrome over, and it's not going to be able to bind to that known antigen. So it's going to be blocking it. So we're not going to see any color. So a positive test is going to have no fluorescence. But if we run our sample, we're always going to be comparing, with a positive and a negative, so a known positive and a known negative. When we do our negative sample, we are going to run our patient's serum. We know that they're negative because this is a control, so this is your control sample. It's going to bind it's not going to be able to bind to these. So this antibody is not going to be able to bind to this. So that's not good news. So if that's the case, then what's going to happen is our ones with the fluorochrome, so our ones with the fluorochrome are then going to be able to bind. And if they do bind, we're going to see fluorescence. So that's kind of wacky because a negative test is going to be fluorescing and a positive test is going to have no fluorescence. So that's kind of interesting. Maybe take note of that because it's different than the rest. But it is cool. I think it's competitive. I'm a competitive person, and so I really appreciate this test. All right, great. Let's go on to the last two. So now what we have is we have a Western blot test, which is pretty cool. So essentially, we have a gel electrophoresis, and so we know that proteins, they're charged, and what happens is we will insert proteins into a lane just here, And what happens is we put it in an electric plate basically, and the proteins will move depending on their charge. And then, so from there, the proteins will move along the lanes, and they'll become bands. And then what we do is, so depending on if we're doing a direct or an indirect Western blot, so direct would be you have an unknown protein antigen, and then an indirect would be you have an unknown antibody. So those are what we're trying to detect. And so the example for the direct, which is the only one that we could find, is for bovine, I think it's spongioencephalopathy, and then the indirect is for FIV. And the reason why we know that this will be positive, if it is positive, is because it's indirect, and we know that FIV for cats, they have it for life. So if we see antibodies, we know that the cat is currently infected with it. They might not be showing symptoms, but we do know that they are actually positive for it. So what happens is we run the lanes, and then from here what we'll do is, depending on if it's direct or if it's indirect, we're going to be running basically the same sample as we did before. So for the direct, it's an unknown protein antigen. So we don't really know what the antigen is going to be, so we're just going to run our fluorochrome over it, and if we see a fluorescence, then we're going to know that it is positive. If we don't, we're going to know that it's negative, and basically the final step will be determining, we'll see an actual color. And the indirect is just, it's going to be an unknown antibody, so it'll be a known antigen. And so what happens is, if we know what the antigen is, we're going to run our patient's serum over that, and then if the patient has an antibody for it, it's going to stick, and then the antibody is going to we're going to run it with the fluorochrome. And then if our patient does have the antibody, we're going to see fluorescence. So that's kind of a cool one. It's really neat. It's charged. Okay, great. So let's go to the last one. Oh gosh. Oh no. All right. So the last one is immunohistochemistry. So what this does is it's going to detect antigen in tissue sections. So it's kind of similar. So we're going to have tissue sections. So this is a tissue section here, and we have, so we're going to start with our positive control, so we know that there's going to be a color change. So basically when we're running these, we have a positive and a negative control, and there's some horseradish stuff in it, which is going to, basically if it's positive, it'll turn brown. We know that it's positive because of that horseradish. And so what happens is these are going to be, we have these unknown antigens, and then we're going to come along basically, and with our patient serum, we're going to run, sorry, I messed that up, so take that back. So you're going to have the tissue section here, which is from our patient, and then we're going to run an antibody over it, and then if the antibody can bind, we're then going to use a specific type of like fluorochrome, and basically it's called chromogen, and it's going to be converted with an enzyme that's on this anti-antibody. And then if we see brown, we're going to know that it's positive, and then if we don't see anything, which we'll just use an irrelevant antibody. So our antibodies are both going to be known for this. So we use an irrelevant antibody that we know 100% is not going to stick to those antigens, so we're not going to see anything here. And examples of this can be a mammary gland tumor in a rabbit. I hope that didn't confuse you. If it did, I'm so sorry. But so that's everything. If you have questions, let me know. Thank you. And I also want to just reference that I used the notes from the immunology lab.